So, Bio 121, we're still in genetics and what did Mendel add to the world and how is this added on to. Somehow we find over the course of time that we don't just look at cows and plants and peas and snapdragons and all that sort of stuff. Eventually we end up looking at what happens when you have many characteristics. And a very good model organism to do that with is actually Drosophila. Drosophila, there's a wild type, and in this case the Antennapedia phenotype you're looking at here, where the mutations are found on the legs and the head in place of antenna, right? So you're going to literally put legs where they don't belong. So how does this happen? These are called homeotic mutants. Let me draw something for you here. You may or may not have realized this already, but there's a basic pattern for all insects. Okay, I'll put some wings on you guys over here. I'll put some legs outside of there. Okay, and the idea, of course, is that when you actually look at their growth properties or what happens to them, right? Let's change color here. Put that in as red. Put a red over it. Okay, you can see that they actually have a pattern, and that pattern is inside the thorax. You have these segmented areas. And what can happen is you can change the expression of what's happening inside each of these homeotic cassettes. And that's why we call those, together we would call these homeobox genes. And what happens is you can make mutations and then express things where they shouldn't be expressed. So that would be one of the sets of legs here. Okay? No, they did not put the antenna on the thorax of the body, but in this organ system, you can begin to look at the many different characteristics that could be changed by examining gene expression inside of tissues. Okay? But it's all based on the same idea. What are the alleles of a gene that's being expressed, and what would it look like? We think of something simple here. Well, it's actually not simple at all. Alleles for drug resistance inside the malaria parasite, not parasite. Sorry about that, guys. Put an E there. Okay, malaria parasite. The mutation that's found here, okay, for here's Anopheles, right, or African malarial mosquito. What it's doing, of course, is it's transmitting malaria-causing parasite into the human. And what's happened, of course, is using false color scanning electron transmission microscopy here, you can see this is the organism here, and what it means is that alleles confer drug resistance to this. Okay, so the alleles of the genes that are expressed on the surface of the malaria is what save it from, wait for it, chloroquine and hydrochloroquine drugs that are causing all the rage for COVID-19 treatments at the time, okay, are what actually work well for malarial parasite stoppage. Now, another way to think about how it is traits can be seen is that in this case, we could link something to the X chromosome or the Y chromosome. And if you take a look at the eyes inside our Drosophila here you can see that patterning system. Look, there's a number of legs, right? And there are the wings, and there are the eyes, and there's the head. But notice some of these guys have brown or red or white eyes. And let's go through clockwise from the top. You have brown, cinnabar, sepia, vermilion, white, and red. Okay, so these are all the distinct color types, okay, and each are dominant to the white color, which means similar to our bunny rabbit experiment a little while ago, you can begin to map how they interact with one another relative to the sex-linked trait on the X chromosome. So here's what we do. We put together a Punnett square with our XX, our XY, and then you see the colors actually coming through, and notice that they transmit. If we're crossing here with a XYW, all female offspring have red eyes, all male offspring have white eyes. So try and do this cross on your own. Take a look at the XWY, XW, XW, lowercase w's here. What you can actually see is how it all bears itself out. So the recessive is what actually causes the white phenotype. How this works in, say, humans. Everybody worries about this sort of stuff, and every textbook explains it poorly. This is one at least decent explanation where they're using numbers that everybody can wrap their brain around. A son of a woman who is a carrier of a recessive X-linked disorder will have a 50% chance of being affected. Now, pay attention to that sentence. The son of a woman who is a carrier of a recessive X-linked disorder, which means the likelihood of actually getting it isn't all that high. However, a daughter will not be affected, but she will have a 50% chance of being a carrier like her mother. So how does this actually happen? Affected, unaffected. Okay? So the son of a woman, okay, would actually have unaffected father, unaffected carrier mother. Okay? And here you can see the unaffected son, but it will have a 50% likelihood of being affected. How does that actually happen? The affected son here is it's on an X-linked chromosome, and it's not carried on Y. And that's literally the explanation. The Y chromosome over here doesn't have the recessive allele. The X-linked chromosome does here. So when you do the crosses, this is what you end up with the four possibilities of actually recombining these genomes to give you a new zygote and therefore new diploid structures. And that's why the son is the only possibility. Another example, okay, are lethal alleles. Lethal alleles are not fun things for people to think about. Lethal actually means they're going to kill you. Some alleles are so non-functional as to kill the individual inheriting it. Manx cats are the classic example, okay? And what it does, of course, is it doesn't actually have to kill it lethally dead. It just has to shorten the normal lifespan. So, all Manx cats are heterozygous for the ML allele, Manx lethal. MM homozygous cats are normal house cats, no problem. ML heterozygotes are Manx. Okay, notice there's no tail here. And then Manx lethal are homozygotes, and they all die. Why? I want you to go away and I want you to think about what this would actually look like. But you already know the answer to the problem. Now, trihybrid crosses, this is the stuff that throws biology students into a whirl. They're like, oh my god, I've got all these different possibilities. And the idea, of course, is if this is a genetics class, you'd be doing these all the time. What I want you to do is I want you to see 3 yellow and 1 green, and then you have 3 round and 1 wrinkled, and then you cross these one more time. Take a look at what actually happens to the output here. When you go away and you look at it, the probability for a shape that occupies the second row, and the probability for the height occupies the third row, the probability for each possible combination is calculated by multiplying the probability of each individual trait. So this actually is not a simple probability problem, but a multiplicative probability problem. Finally, as we move into what's actually happening with chromosomal crossovers, this brings us all the way back to the end of chapter 12. This is why I said I thought it was actually sort of really bad form that your textbook just throws this in at the end without teaching you how this could have happened. So how we actually get crossover events to occur and segregations are through those meiotic steps. And remember, in prophase 1, we form those tetrads, and those tetrads now will cause the recombination of this genetic information. And here's what I want you to look at. Large A, large B, large C. Large A, large B, large C. Small a, small b, small c. And you can see the pattern. Once recombination has occurred, take a look at what's actually happened here. Large A, small b, large A, large B, large C. Large A, large B, small c. And you can see the pattern. And now if we did crosses amongst any of these trihybrids, you could now begin to see what's happening here. And here's the reality. This could happen anywhere on any of our 23 pair of chromosomes. So the reality is that during meiosis, for any one of your sets of chromosomes, for 23,000 genes, you then change the likelihood of actually ever seeing any meiotic output. And that's truly what makes you and your life amazing. But we don't tend to go into that in this class. Now, what's going to happen, of course, is that even though you have this entire possibility of having these genes, we have one more consideration to take under our observation, and that is epistasis events, where genes are not independent, but rather interact to produce phenotypes. So a moment ago, I mentioned coat color when we were talking about, say, rabbits or mice or something like that. And you have this model, what we call a Goody coat color. And its A is dominant to the solid coloration of, say, black or gray. But the gene is at a separate locus for C, is responsible for pigmentation. And the recessive C allele does not produce pigment. So you can see how we can get albino mice down here. There has to be a reason why this would happen. So a recessive C allows for the albino that you see. A mouse with a homozygous recessive CC genotype is albino regardless of the allele present at the A locus. Take a look up here. Small c, small c. Set up your crosses. Large A, large C, small a, large C, large A, small c, with small a, small c. When you do the cross, you can see this is actually how we end up getting our recessive phenotype being expressed independent of the dominant allele, which was A. Okay? Such multiple interactions produce modified Mendelian dihybrid crosses, where here now the ratio is 9 to 4 to 3. And this explains sometimes why it is that inside a family you might have a relative that has a completely different genotype than what you see for the rest of the family members. Okay? This is big news back when this was discovered, and it was only the genetic mechanism was only measured. It wasn't all that long ago. It has to be 10 years ago or something. No, it's more than 10 years ago now. I've been here for 15 years, so it's 16 years ago, maybe 20. Okay? So this is actually a fairly new idea. I mean, not a new idea. It's been observed in biology for years. The mechanism has now just been worked out. Then again, I'm getting old, so new for me might be like, wow, that's like way old for you guys. Okay, so that's chapter 12, Bio 121. Hopefully you've paid attention. You've paid attention closely. You can see the details are getting a little bit more wild now as we go along.